just going to wait for this to load. Okay, there it is. So when we talk about organelles, I know when I taught freshman biology, I tend to just teach them separately, but really we want to look at it as a more holistic or inclusive approach. So just being refreshing yourself with the basic functions was good, but now we kind of want to look at it from an upper level point of view. And so this is part one of how certain organelles work together for a common purpose or function, and that is going to be the endomembrane system. So if you learn nothing else in AP biology, you should learn that proteins are probably the most important thing in any organism. And so for daily life processes and growth, the cell needs to read the genes, which are small sections of DNA that code for a particular protein. We need to then build those proteins or synthesize them. And those proteins are used in a variety of ways, including but not limited to structural proteins that form the muscles and fibers and hair, skin, claws of organisms. Proteins are enzymes, which are molecules that speed up chemical reactions. And we're going to have like an entire unit on just enzymes. And proteins can be hormones, and they also can be receptors. So remember, hormone is an umbrella term for any chemical messenger that's produced in one part of the body and then travels through the bloodstream to another part of the body where it then has an effect. Now, steroids are lipid-based hormones, which means they can slide right between the phospholipids and go right into the nucleus and switch on genes. Some signals are protein-based, like insulin, and so it will then need to bind to a protein receptor on the cell membrane. So you've got a protein signal binding to a protein receptor. So it's all about proteins. So the nucleus is the central organelle of the cell. It is made up of a nuclear envelope that has pores. Those pores are going to allow RNA to leave the nucleus and head out to the cytoplasm. There's also the nucleolus, which is this darker round structure here, and the nucleolus creates ribosomes. And then finally, inside the nucleus is chromatin, which is a network of DNA and proteins. So scientists can't keep things simple. And so basically, we know that DNA is in the nucleus, but depending on what stage of the cell cycle a cell might be in, the DNA can take on different forms, and so it gets different names. So chromatin is when it's just completely unorganized. It's not, stacking up to form chromosomes, and it just kind of looks like a big plate of spaghetti noodles. Chromosomes are when the DNA begins to wrap around proteins and stack right before nuclear division during mitosis. Then when you have the original chromosome and its copy in the shape of an X, we call those sister chromatids. So we'll sort all that out later. Just know that it's all, no matter what, it's always DNA. It just kind of is organized or not organized. The function of the nucleus is to basically house and protect the DNA, which are the instructions for building proteins. DNA can never, ever, 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 ever leave the nucleus, so it always stays home. Next, we have the ribosomes. The ribosomes are incredibly tiny on the scale of cell size, and they can. One thing you may not have learned in Biology 1, as far as their structures, they're made up of two subunits, a large subunit and a small subunit. And so the large subunit in the diagram is that big orange blob, and then the small subunit is the kind of bluish thing underneath. Ribosomes can be free floaters, or they can be attached to the rough ER. 60% of the ribosome is ribosomal or rRNA. Can you guess what the other 40% might be? You're right, it's protein. The function of the ribosome is basically protein synthesis. So we're going to build a polypeptide chain, the primary structure, according to the directions encoded in messenger RNA. And the central dogma of biology is this idea that DNA has to be rewritten into a form that can leave the nucleus, which is messenger RNA, that then is translated into the language of protein. And so the central dogma is just a tenet or an important feature of biology. The endoplasmic reticulum, or the ER, is a series of folded membranes. There's two kinds. There's rough ER, which has ribosomes attached to it, and there's smooth ER, which does not. The interior of the folded membrane that kind of, looks like a trough in this cross section or a ditch, we refer to that as the lumen. The function of rough ER is to transport proteins. We kind of always say that ER is transports proteins, but really now in AP, we're going to kind of differentiate between rough and smooth. So rough is protein transport. Smooth has three functions: to synthesize steroid hormones. Remember, steroid hormones are lipid base. They can store calcium ions in muscle cells and detoxify drugs and poisons. And then we need to address the lumen versus the cisternae for when we kind of talk about this process as a whole. So the lumen is the inner space inside the Golgi or the ER, and the cisternae is each compartment or folded section. And so you might want to write this down or type this in the box that just says endoplasmic reticulum, because I didn't include a picture because it was already in the picture with ribosomes and that would be redundant. But we definitely have to know the difference between lumen and cisternae. The Golgi is also a system of folded membranes and vesicles. The cisternae are the Golgi compartments, and the cisternae has two sides or faces. The cis face is the receiving side, so this will be the side that would be facing the endoplasmic reticulum, the ER, because it's going to receive a little vesicle, kind of like a rough draft protein, and then it's going to put the finishing touches on it. The trans face is the shipping side, so that side's going to face the cell membrane because that's kind of like the last stop before a protein or other cellular material is exported out of a cell. The function of the Golgi is to ensure correct folding of secondary, tertiary, and quaternary structures and add chemical modifications to newly synthesized proteins as needed. And they're going to package and distribute, sorry, I can't talk, those proteins to either within the cell or to the cell surface at the plasma membrane for cellular export. All right, that's it. The end.